Okay, so we're going to continue chapter 2 in this video. So the purpose of the cell membrane is to keep the outside outside and the inside inside. So if you have constant mixing between the outside and the inside, then you're not alive anymore. That's one of the first necessary things for the first cell to evolve, is to make sure that that the stuff on the inside stays inside and all the things that are on the outside stay outside. However, you do need to eat and you need to get rid of the waste products. So things still need to cross the membrane to get into the cell, and things need to cross the membrane to get out of the cell so you don't accumulate waste products. So how does that work? How does your waste product or nutrients move in or out? So there are three kinds of processes that do not require energy to move things. And this is called passive processes. And there's going to be one called diffusion, osmosis, and then facilitated diffusion. Okay. So this is called, first of all, passive processes because it does not require energy. It does not require ATP, which is the currency of energy. This is called in this case it's a physics, and maybe you reviewed this from General Bio, what's diffusion, and what happens with diffusion. So diffusion happens with any kind of molecule. Any kind of molecule, if they're small enough, they can just diffuse through the membrane. Something like water, oxygen, or carbon dioxide, they will just diffuse through. Whereas osmosis, I want you to remember that this term only applies to water, this movement of water. So only water is moving from one place to the other. And then the facilitated diffusion means you've got something that's too big, and this molecule is too big to cross, basically has a charge, or the size is too big. It's not going to go through the membrane, and you need help to facilitate for it to go through the membrane. How do you help? You put a protein here. So protein has a tunnel, and there is a space where the charged molecule make its way inside of the cell, and it does not require energy. It just requires to have a cell that's the right shape and things like proteins for it to pass through. So that's passive processes. Then we have active processes. So the word active means it's going to require energy. So I'm going to say requires energy, or ATP, which is the energy currency. So you can see that, there's ATP here. You can clearly see that, and that's helping. It's a protein. So there's a protein enzyme, and then there's ATP that changes shape of the enzyme, which causes it to grab a molecule from here. It will grab a molecule from here and transport it to the other side. So if you're blowing a balloon, you take energy to blow air into the balloon. If you didn't put energy into it, the air will not go into the balloon. So in this case, whatever this molecule is, it doesn't want to go inside the cell, but the ATP is forcing it to go inside the cell. So that's the one type of active transport. Another type of active transport is, or process is, where something goes into the cell membrane and the cell membrane wraps around it. So here, so something goes into the cell membrane and the membrane starts to kind of wrap around it, and eventually it's going to be wrapped around it completely. And this is called endocytosis. So what does the word cyto mean? It means cell, and then cytosis means cellular process. So this is a cellular process where either like food or some sort of liquid that gets absorbed into the membrane, and the membrane wraps around it, forms a bubble, and then it goes into the cell like this. And once it goes into the cell, then the name of it, then it can get digested, and it can be called vesicle. And once it goes into the cell, it gets digested. Then when it gets digested, it turns into waste product, and then that waste product needs to be excreted out. So they leave the cell. That's the opposite of endocytosis. That's called exocytosis. So now we have all your waste products, and those waste products are wrapped around the bubble, and this is called the vesicle. And the vesicle will merge mid the membrane and dump all the stuff, all the waste products outside, and that's exocytosis. So now going over here, endocytosis has two subcategories called pinocytosis and phagocytosis. So the word phago here means to eat. So phago means to eat. And then the word pino means to drink. So phagocytosis is where you eat something large. So phago means to eat. Maybe if you're an amoeba or a white blood cell, you gobble up like a bacteria, that's going to be called phagocytosis, eating something really big. On the other hand, we've gotten this other term, pinocytosis. So pino doesn't mean to eat, it means to drink. So maybe amoeba or a blood cell, they drink some liquid and then they go ahead and do pinocytosis, where they wrap around the cell membrane and they bring the liquid into the cell to get the liquid on the inside. So that's pinocytosis and phagocytosis. Okay, so cell membrane is not always going to be flat. And so you have some modifications to the cell membrane as well. If you go ahead and maybe give it some extra surface area, then, maybe, so this is flat cell membrane, but if you go ahead and give it some extra surface area like this, and these are going to give it some extra surface area, but these are called microvilli. Villi stands for fingers. So these are little micro stands for small. So these are little microscopic finger-like projections that you have on the surface of the cell membrane. So finger-like projections of plasma membrane. Not all your cells are going to have this, so it will exist in some tissues that require you to absorb material. So maybe you can think of a place in your body where you might want to absorb things. So maybe in the digestive system, you'll find microvilli in your intestines. So you'll find maybe in your kidneys where it needs to go ahead and absorb fluids. So this is going to, the microvilli is going to increase the surface area. And that's going to help you absorb or get rid of stuff. And the whole point of microvilli is that all you're going, so the microvilli increases surface area, but it's not needed everywhere. So it's not going to exist on all of the plasma membrane or in your body. It's only going to depend on what tissue you're talking about, which have microvilli, which might not, some might not. So then this is a generic cell that's showing that it does have a microvilli here. If this was flat, then there would be no microvilli. So we're going to talk about a few other things. But before we talked about this, I guess, highlighted stuff, let's go ahead and talk about cytoskeleton, or the cytoplasm here. So since we talked about cell membrane, what about the inside the cytoplasm? So we said that there's cytosol, that is the fluid, that was the fluid, and the intracellular fluid is going to contain your dissolved chemicals, your nutrients, your glucose, your proteins, the waste products. So all that is dissolved in this fluid, cytosol. And ICF stands for intracellular fluid within the cell. And then you also have the organelles. So the intracellular structures that perform specific functions. These are the organelles, the mitochondria, Golgi apparatus, endoplasmic reticulum. So we'll go through some of the really important ones right now. Okay, so we have some stuff highlighted around the nucleus, but I think I said before, we're gonna go over the cytoskeleton first. So cytoskeleton is, maybe tell me what cyto stands for. And if I was in person, some of you would tell me that stands for cell. So skeleton means there's a skeleton inside the cell. So this would basically be the kind of like scaffolding or the framework inside the cell that exists. That holds things up in place. And you can see over here that you got organelles in between. There's cytoskeleton that's holding all the organelles in place. And cytoskeleton is it's not going to have a membrane. So there's non-membranous organelle. So instead of having a membrane, the cytoskeleton is going to be made up of protein fibers. And these protein fibers are going to be named based on how thick they are. So if they're super thin, they're called microfilaments. If they're going to be really thick, then they're called thick filaments. So it kind of depends on the thickness of the filaments, and that's how they're named. And different kinds of thickness in fibers and then you combine them and they're able to form a cytoskeleton. So you can see here the sizes: microfilaments, microtubules, intermediate filaments, and microfilaments and microtubules here as well. So again, the function of cytoskeleton is to hold things in place, but that's not the only function of this protein. So sometimes this protein is used to move stuff around. So if you think about, again, general biology, and you think about the process of mitosis and meiosis that you learned, there was a phase called prophase. So, there were four different phases, but there was also something called prophase that had microtubules that grew and like this. And so these are something called centrioles. So this thing called centrosome, this is a microtubule that grows and grabs onto chromosomes. They push and pull on the chromosomes until they can make it to the right part of the cell. So you need to have this in order for cell division to occur. Now, one interesting thing is that this happens in the human body, but your nerve cells don't have centrioles. So if you don't have centrioles, you can't make microtubules. If you can't make microtubules, then you can't have cell division. This is the reason why your nerves have such a hard time repairing themselves. And they might not be able to repair themselves at all because they don't have the ability to go through mitosis, or at least most of them don't. So that's why, because they're missing, the reason is because they're missing this organelle called centriole. So in this case, the component here is made up of same material as the cytoskeleton. So this is centrioles are located in the centrosome, produce microtubules that move chromosomes during their cell reproduction. In addition to this component, it can also be turned into something called cilia. Cilia are going to be these little short hairs that move. And if you recall from general biology, you might have this little organism called, we look at it under the microscope, it's called paramecium. On the surface of this cell, it has little hairs that will beat and move, cause paramecium to swim through the water. So we're not paramecium. So instead of having it, on the surface, we have this on the surface of the cells. And on the surface of the cells, you've got mucus here. So you have the cilia, and then there is mucus. Let's say mucus here, and that's going to be more. That's the cilia is going to help move the mucus. So, and then there is this, maybe small bacteria in this mucus that's also going to be being moved, and it's going to move past the cells into your throat. So this picture is showing right here. It's going to be moved into your throat, and then it's going to go into your stomach, and that's where you have the stomach acid that's going to sterilize and going to kill these little microorganisms that get trapped here. So the function here is not to have paramecium swim through the water, but to move things past your cells. In this case, it's going to move fluid secretions past your cells to go somewhere else, and then to another place when you, so another place you have this happening is in the fallopian tubes. So that's how the eggs get moved to from one end to the other end in the fallopian tubes. So this is located at the cell surface. They're anchored by basal, and then body just under the cell surface, and then the beat rhythmically to move fluid or secretions across its surface. And the name of this tissue is pseudostratified ciliated columnar epithelium. Is referring to this tissue here. Okay, so what if you take one of these cilia and grow it really long? Well, it's not cilia anymore. Now it's going to be called the flagella. And I know you want to call this tail, but please don't call it tail in anatomy. We're trying to teach you anatomical terms. So if I were you, maybe put a picture on a, I were to give you a picture on a quiz and point to the flagella and ask you to write down what this is. Don't write down tail, because I'm going to mark you off wrong for that. It's because you didn't write down the anatomical term, and that is flagella. And so this will be, found on the cell surface as well, and helps the sperm swim through the fluid. So if you remember from the previous picture, there are hundreds of cilia, and they're really short hairs. They're almost like carpet of cilia. They're moving all together. On the other hand, flagella is only found in the sperm. That's the only one. There's only one flagella per sperm, and some sperm do not do well 2 or 3 flagella, but those are not functional. So healthy sperm has only one flagella. And if you do a cross section of it, it's a little bit more complex than cilia, but it's still made up of microtubules. It has ATP or energy because it has to move, and this will cause the sperm to propel through the fluid. And the other organisms, like, I don't know if you remember dinoflagellates, learning about dinoflagellates, which is a phytoplankton. It's called dinoflagellates because it has 2 flagella. So flagellates refers to flagella. So in the organism, there's 2 flagella, and they are used to move up and down in the water column. But we're not talking about dinoflagellates. We're talking about humans. In humans, there's flagella on the sperm that's going to help it swim through the fluid. Okay, so next non-membranous organelle we have is called the ribosome. So it's a little molecule that makes proteins, and there are 2 kinds that you got. There are free ribosomes that are just floating around, and then there are ribosomes that are associated with the rough endoplasmic reticulum. Ribosomes make proteins, so they're cells' protein factories. So this is something that's called this, that makes this rough endoplasmic reticulum, because it makes the appearance kind of looks rough when the ribosomes are attached there. You guys probably learned about this in general bio about translation and transcription. You learned that DNA goes to RNA, and then RNA becomes the protein in the translation and transcription process. So you have to send this RNA into the ribosome in order to make that protein. In fact, the ribosome here is the actual RNA. So let me write that down. It's rRNA. I don't know if you remember 3 types of RNA. There was mRNA, tRNA, and this is rRNA. So they are involved in making proteins, and but the nucleotide is so that's a nucleotide. RNA is a nucleotide, but the nucleotide has configured to work and as an enzyme, and it's basically a little kind of chemical machine where you run the information through the mRNA and then outputs on the other end and gets a chain of amino acids through rRNA. So here is what they look like. This is showing a bunch of little dots, and that's what free ribosomes look like. And going back to transcription, translation, in this case specifically translation, there's going to be the protein production basically through this. And we don't have a microscope that's small enough to see these little dots. So that's the nucleus. These are just free ribosomes that look like dots floating around. This would be the endoplasmic reticulum. It has one large subunit and one small subunit, and basically it kind of works like a little machine where it's going to feed into this, and then it's going to output on the other end a chain of amino acids. So this is the so basically you have two subunits, small subunit. This is like I tried to say this is like a little house, and that's basically you're feeding something into it, and it's a protein that's coming out. So mRNA goes through it and outputs on the other side a string of amino acids that we call protein. So this is going to do it for the non-membranous organelles. So next we have the membranous organelles. So I'm going to stop this video here, and then I'll make another one for the membranous organelles.